In this video, we are going over literally every single amino acid, and if you're familiar with bone broth health benefits or collagen, then you probably already know what glycine is. This is the first amino acid we're going to go over because it is so simple, super easy to understand. So first of all, what even is an amino acid? An amino acid, there's 20 of them like that are biologically relevant to you and your health. An amino acid is any of these molecules that have a central alpha carbon, an amine group here, and then a carboxylic acid group here. So, and then a side chain. And then there's 20 amino acids that we're going over today. So see how they all literally have that. They have the amine group, the alpha carbon, the hydrogen sticking off the carboxylic acid group. And then what makes every single amino acid unique biologically, like what gives it its specific function in health, in metabolism, in biochemistry, is the fancy group sticking off here. So isoleucine, this is a branched chain amino acid. We say it's a branched chain amino acid because here's the amino acid, and then here's the functional group, and it's branched. This is glycine, and it has an H sticking off of this alpha carbon. Next amino acid is alanine. Pretty much alanine and glycine are the exact same thing, except in alanine, instead of the functional group being just a hydrogen atom, we have a methyl group, so CH3. Glycine has a hydrogen, alanine has a methyl group. This is isoleucine. This is a branched chain amino acid. It's a BCAA because here's the amino, here's the functional group, and its chain is literally branched. Similarly, we have leucine, also a branched chain amino acid because the chain is branched. Also, we have valine, another branched chain amino acid because its chain is literally branched. So valine, leucine, isoleucine, these three fellows are your branched chain amino acids because they have branched chains. This is methionine. Methionine is literally K, so here's the amino group, here's the carboxylic acid, and then here's its long chain, and it's got a sulfur atom here, and then off that sulfur atom, it's just got a methyl group sticking out. So this is methionine. This is proline. Proline is really weird because it bonds with itself. So here's your amine group, NH2. Here's your carboxylic acid group. Here's your alpha carbon with the hydrogen projecting out. And then proline's functional group projects out and around itself and then covalently bonds with its amine group. So now because this nitrogen is making 4 bonds, this nitrogen has a positive charge right here, but proline is still hydrophobic and it's still non-polar because this functional group here is hydrophobic and non-polar. All these brown amino acids, these are hydrophobic, non-polar, and uncharged. So your branched chain amino acids, your BCAAs, these are hydrophobic. They do not want to associate with water. They do not have any charge, and they are non-polar molecules. Next group of amino acids, these are your aromatic amino acids, and I've switched up the drawing convention a little bit to further better show you what amino acids actually chemically look like. Here is your alpha carbon, here's your amine group, and then right here, this funny setup, that's your carboxylic acid group. So this is your COOH. Recall from the first few amino acids, all I did was write COOH. When you actually draw that carboxylic acid chemically, it looks like this. Pretend there's a carbon right here, and then a carbonyl group, so a double bonded O, and then a hydroxyl group and OH. So COOH, that's your carboxylic acid. This amino acid is tryptophan. I don't know if I can say this or not. I remember tryptophan because I think tryptophan looks like a snowman with pointy boobs. Like, this is the snowman's body, and then this is like its pointy boob. It is aromatic because we have two aromatic groups here, and then it's got an NH group sticking off, and that's the snowman with pointy boobs. Next one is phenylalanine. If you guys know what PKU is, phenylketonuria, it's like a metabolic disorder where you cannot metabolize phenylalanine. This is the amino acid phenylalanine. It's aromatic because it's got this big aromatic group. And then if you know what phenylalanine is, it's super easy to know what tyrosine is, because tyrosine is literally phenylalanine, but just with the hydroxyl group on top of the ring. So phenylalanine is non-polar, hydrophobic, no charge. Tyrosine is polar a little bit, hydrophobic a little bit, still doesn't have a charge, but what we have going on here is we have a hydrophobic, non-polar amino acid, but then we've given it an OH group right here, a hydroxyl group, and hydroxyls, OH, are polar, and they are hydrophilic. So most of this thing is non-polar. This big, large aromatic ring is non-polar, but now that it has this OH group sticking off, it is slightly polar. So this hydroxyl group has potential to make hydrogen bonds or other hydrophilic interactions, but this big, large ring here, and then this carbon with two hydrogens sticking off, makes it hydrophobic. And if you know what phenylalanine is, you know what tyrosine is, because all you have to do is add that hydroxyl group. Next class of amino acids, these are your basic amino acids. You can just imagine that these amino acids are sipping on Starbucks and they're wearing Ugg boots and black Lululemon leggings because they are basic. So here is histidine. It is a ring, and then it's got a nitrogen here and an NH here. And histidine can become protonated and be positively charged. So this NH group here can either be NH or protonated into NH2, and then this histidine is going to have a positive charge and it's going to be basic. This is arginine. Where the frick did lysine go? This is lysine and this is arginine. So let's start with lysine first. Here is the main amino acid situation. The amino group, the carboxylic acid, alpha carbon with a hydrogen. And then to know specifically what lysine is, how I remember lysine is you literally just go 1, 2, 3, 4, NH3 plus. Because this is a nitrogen with 3 hydrogens attached, it is positively charged. And so lysine, because of its functional group having a positive charge, it is basic. It can donate a proton. So this is CH2, 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 NH3 plus lysine. Arginine is a petite little bit similar to lysine. Ew, gross hair in my mouth. Arginine, petite little bit similar to lysine, except it's got a lot more going on down here. So here's your main amino acid situation, and here is arginine's functional group. So CH2, 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 NH, C, NH3, NH2, NH2 plus, NH2. So let's go over this really quickly. When you have NH2 or an NH3, when you have a nitrogen atom, that nitrogen atom likes to make 3 bonds. So right here we have NH2 bonded to a carbon. So this is nitrogen, hydrogen, hydrogen. Then one nitrogen atom is holding on to 3 things, 1 carbon and 2 hydrogens. Down here, the nitrogen atom is holding on to 3 things. It's making a double bond with this carbon, and then it's also holding on to 2 hydrogens. So when a nitrogen atom is making 4 bonds, that nitrogen has a positive charge. Whoa, this is a messy situation now because of the wind. Let's figure out where the frick lysine just went. Here's lysine. NH2 bonded to a carbon, so this is nitrogen, hydrogen, hydrogen, carbon. NH3 bonded to a carbon, so this is nitrogen, hydrogen, 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 carbon. Here the nitrogen has 4 bonds, here the nitrogen has 3 bonds. Because this nitrogen has 4 bonds, it's got that positive charge. This nitrogen only has 3 bonds, so it's neutral, no charge. So that's kind of what is causing the nitrogen atom to have the charge, or the amino acid's functional group to have a charge. Throwing it back to arginine, wherever we left off, at its functional group, NH, carbon, NH3 plus NH2. This is arginine. It is a basic amino acid. On the contrary, these are your acidic amino acids, and they are acidic amino acids because their functional groups are capable of releasing protons into solution. As soon as they start releasing those protons into the solution, the proton concentration increases, and now you are acidifying the solution. So we have 2 acidic amino acids. Those are glutamate, or glutamic acid, and aspartate, also known as aspartic acid. Here is glutamate, here's the amino acid situation, and then here is its functional group. And the functional group is what gives the amino acids its specific role in health, biology, biochemistry, metabolism, everything. The functional group is what gives it its function. So here we have glutamate's functional group, CH2, CH2, and then a carboxylic acid. See how this is a carbonyl and then an O negative? We're gonna call this glutamate. If this was OH, it would be glutamic acid, because the hydrogen atom here, because it's an OH, it has potential to release that H into solution and acidify the solution. The way I've drawn this amino acid, it's already released that proton into solution. It has a negative charge now, and it is glutamate. If this was glutamic acid, this would be OH. So this is a carboxylic acid. Just like this is COOH, this is COO, and then there was an H, but it acted as an acid and it released that H, that proton, into solution. Aspartate, aspartic acid, is pretty much the exact same as glutamate, except it is 1 CH2 shorter. So we have aspartate, the amino acid here situation, and then its functional group is CH2, carbonyl, and then OH, or because it has acted as an acid, it's disassociated that proton, released the proton into solution. It is now existing as aspartate, and this O has a negative charge. Last little collection of amino acids. These are polar hydrophilic amino acids, and I've drawn them a little differently again. I've drawn them without showing the alpha carbon, and then I've fully shown the carboxylic acid group, the amine group, and then the functional group. So let's go over this really quickly. Here's your alpha carbon, here's your carboxylic acid, here's your amino group, and then all this here is the functional group. So this is cysteine, and its functional group is CH2SH. So here's a sulfur atom bonded to a hydrogen. Cysteine is super critical to the glutathione antioxidant system because of this SH situation. So right now, because it's SH, this is cysteine reduced. If it were to be just S, it would be cysteine oxidized. And that difference between cysteine reduced and cysteine oxidized is absolutely central to the function of the antioxidant system glutathione. So this amino acid, so critical to brain health, cellular health, mitochondrial health, longevity, because of its role in that antioxidant system. And this is all coming down to its functional group. Like this part here, makes cysteine unique, and it's what makes cysteine actually able to function in its functional role in biology. Like, the functional group is everything. You do not have biological function if you do not have that functional group. Next one is threonine. Threonine is threonine, cysteine, and serine. I always confuse. So cysteine with the SH group. I'll show you serine right now. Serine is exactly the same as cysteine, except instead of the SH group, it's got an OH. And I also screwed up this. This should be CNH2, not carboxylic acid NH2. So this is screwed up. This is essentially what serine is. It's an amino acid and its functional group is CH2OH. Threonine, it's kind of similar to valine in that its functional group is branched, but right here it's got an OH group. So this is threonine and it can make hydrogen bonds and it is polar. Here is asparagine. Here's your carboxylic acid. Here's your alpha carbon, here's your amine group, and then CH2, carbonyl, NH2. This is asparagine. Last one, glutamine. This is glutamine. Carboxylic acid, alpha carbon, NH2, CH2, CH2, carbonyl group, NH2. So glutamine and asparagine are very similar, and if you're interested in amino acid metabolism, amino acid synthesis, or the urea cycle, you will see glutamine, asparagine, glutamate, aspartate. You will see those amino acids show up all over the place, and it is because glutamine and asparagine are so ridiculously similar to aspartate and glutamate. They are very easily metabolized into each other. Your body can make them from one another down these different metabolic pathways. That was like ultra high level overview of literally every single amino acids. Like, comment, and subscribe, and let me know in the comments like what went well in this video, and then tell me as well what I need to do better. I make these videos for you. I want to share this biochemistry stuff that makes me excited, but I know I'm not like very good at sharing it yet. So let me know in the comments how I can make this better. Okay, see you later, everybody.